So our next speaker is Dr. Hui Xin Ong, and some of you may know her as YY. And YY is a research leader at the Woolcock Institute of Medical Research in the Respiratory Technology Group and a senior research fellow at Macquarie Medical School. So with a PhD in pharmaceutical science from University of Sydney, her research is focused on developing pharmaceutical and device technologies that can be used for the treatment of respiratory and infectious disease and methodological platforms that can help to promote the translation of new discoveries to clinic. So let's hear from YY to talk about mRNA delivery.
Thank you for that very kind introduction. Good afternoon, everyone. So today I'll be talking to you about a project that Ab Initio has been performing together with therapeutics in enhancing the intranasal delivery of mRNA therapeutics using a novel thermosensitive polymer called tetramatrix polymer. So briefly, for some background, RNA-based therapeutics such as mRNA, siRNA, oligonucleotides are disruptive technology that is rapidly expanding since the COVID-19 pandemic, which will continue to change the standard of care for many diseases. However, current RNA therapeutics that are marketed in the market have only been delivered via intravenous route. So naturally, nasal delivery of this therapeutic RNA presents a significant opportunity, yet remains an underexplored modality to treat many diseases locally in the nose, for example, vaccinations or for flu, or directly to the brain via the olfactory epithelium to treat diseases such as brain tumors or neurodegenerative diseases. So delivering these biologics to the nose has several advantages. Firstly, there are a non-invasive, needle-free approach that allows it to be easy to self-administer. It also avoids first-pass metabolism and bypasses the blood-brain barrier, minimizing systemic side effects and provide a quick onset of action. So collectively, it also helps improve the patient compliance to the therapy. However, there are several challenges to wanting to deliver these biologics to the nose. There are the rapid mucous ciliary clearance due to the cilia that is in the nose that limits the residence time of this therapy in the nasal cavity. Current devices are also have poor nasal regional targeting, where the drugs are also subjected to enzymatic degradation in the mucus and coupled by low cellular permeability of this large molecule across the nasal epithelial cells. There is also the limited volume of solution that can be delivered into the small area of the nasal cavity. So a common approach to solve some of these challenges includes the use of thickening polymer, such as microcrystalline cellulose, that increases the viscosity of a formulation that enhances mucoadhesivity and hence residence time in the nasal cavity while reducing runoff to the back of the throat or to the front of the nose. But the trade-off for using such thickening polymer is that it is very difficult to aerosolize, which generate either large aerosols or even low-quality aerosols. Therefore, there is an imperative need to develop new excipients as vehicle to deliver these RNAs either alone or as part of a nanoparticle system that has improved mucoadhesivity, generate fine aerosols, and also are able to overcome these biological barriers in the nose. So in this study, we propose to use a unique thermosensitive polymer for delivering mRNAs to the nose called tetramatrix polymer, also known as TP. It has a unique behavior whereby at lower temperature, it behaves like water, which allows for easy aerosolization, but when it hits the nose, because of the high temperature, it then jellifies. This particular polymer has 4 different components which have different functionalities, which includes oegma for its water solubility, NIPAM for its thermoresponsiveness, PLA-HEMA for its adhesivity, and the NAS functional group that allows it to interact with the biological molecule. Now, specifically, we didn't only want to look at TP as a thickening polymer that sticks in the nose, but potentially it could also be used to encapsulate the therapeutic within a nanoparticles system. So as a proof of concept study, we then utilized an mRNA that expresses for green EGFP protein as a model protein. We also then developed a functional assay using rPMI nasal cells to measure the efficacy of this mRNA mRNA formulations, coupled with time-lapse microscope to evaluate the expression of this green fluorescent protein over 48 hours. Now, what we found was that with mRNA alone or mRNA with the TP polymer alone, the mRNA was unable to have a functional effect. But when this mRNA is mixed with lipofectamine, which is a cationic lipid, the mRNA became functional, where we saw an increase in this GFP expression and starts to plateau off at 24 hours. So mRNA needs to be delivered with another cationic lipid to enable the initial cell penetration. So based on these preliminary results, then we designed 3 different formulations to test our hypothesis to see if TP polymer can be used as part of a nanoparticle carrier system. The 3 formulations include TP0 formulation, where mRNA is mixed with lipofectamine only, TP25 and TP35 formulation, where mRNA is mixed with lipofectamine with either 25 or 35 milligrams per ml of the TP polymer. So then we designed a parallel study using a transwell system where we could simultaneously evaluate mRNA functionality and nanoparticle generation and release from the TP polymer. So briefly, the TP formulations were added to the apical chamber of the transwell system, where in one set of plate we have rPMI cells cultured at the basolateral chamber to look at EGFP expression. Then a second set of plate is set up simultaneously with exactly the same conditions, but without any cells at the bottom to look at nanoparticle generation. After 24 hours incubation at 37 degrees, the transwell is then removed, and the plate containing the cell were then taken to the fluorescent plate reader to look at EGFP expression, and the other plate, the medium, is taken to the zetasizer to look at generation of this nanoparticle. Now, what we found was the formulation containing the TP forms nanoparticles of approximately 40 nanometers in size, while the TP0 did not generate any detectable nanoparticles. In terms of the mRNA functionality, what we saw was that TP25 and TP35 formulations showed a significant increase in the GFP expression compared to the negative control and was similar to leveled as a positive control. But the TP0 formulation did not show any significant difference to the negative control, which could be potentially because there was no detectable nanoparticle for that particular formulation or that the mRNA is interacting with the transwell membrane. But nevertheless, what we show was TP can release the functional nanoparticles that is capable to induce maximal efficacy of the mRNA in rPMI nasal cells. We then looked at the stability of these mRNA nanoparticles in terms of mRNA encapsulation. So we measured encapsulation using a ribogreen dye that measures total mRNA and free mRNA. The TP formulations were stored either in the fridge or at room temperature and were analyzed at day 0 and day 7. All formulations were found to be stable in the fridge. However, only formulations containing the TP were stable at room temperature up to 7 days. TP0 saw a significant decrease in the percentage of mRNA encapsulated at room temperature after 7 days. We then evaluated the possibility of using the TP formulation for nasal drug delivery in terms of its sprayability and in vitro nasal deposition. So the formulations were aerosolized using Aptar Bivex nasal spray device, and the aerosol particle size distribution were measured using an inline laser diffraction method. In general, the formulations containing the TP polymer generated larger aerosols compared to TP0. Nevertheless, the formulations are appropriate for nasal delivery as most of the particles were above 10 microns in size and had similar profiles to TP0 formulation. Then we looked at the coverage of this TP formulation in a nasal cast model that is coated with Sargel, which is a moisture indicator. The formulations are loaded into the Bivex nasal spray device and actuated once into each nostril at a 45-degree angle. Pictures of the deposition was then taken every 5 minutes for 20 minutes. So the videos you can see here is taken when the formulations were actuated into the nasal cast model. what we see is that the TP0 formulation immediately pulls to the tip of the nasal cavity, where we saw nasal dripping of approximately 100 microliters that could be collected once the device is removed. This was not observed with the TP25 or the TP35 formulation. Finally, then we use ImageJ to qualitatively analyze the area coverage of the TP formulations, which is represented in white color. Now, what we see is that TP0 had a more slower coverage of the nasal cavity, which could be due to the initial significant loss of the formulation due to the nasal dripping at the beginning. With TP25, we saw a more rapid coverage at 5 minutes, but we also saw some throat dripping at 20 minutes. Finally, with TP35, we saw area coverage at 10 minutes without any observable nasal dripping or even throat dripping. So in conclusion, we've managed to demonstrate that the TP polymer is a promising potential as an alternative excipient for mRNA delivery to the nose. We have showed that it improves delivery to nasal cells with enhanced nanoparticle stability at room temperature, and also offers greater retention in vitro. Thank you for your attention, and I'm happy to take any questions that you have.
Thank you, YY, for the very interesting talk. So do we have questions from the floor? Maybe we'll start with a question from Slido. So I can explain, how do you check the stability of your nanoparticles?
Sorry?
Can you just clarify, how do you check the stability of your nanoparticles?
How do we check? We looked at the encapsulation of the mRNA. So we use ribogreen dye to evaluate or measure the free mRNA. So we did a total mRNA count first, and then minus off what is the free mRNA that gives us, and then divided it by the total mRNA that gives us percentage of encapsulation. That's how we evaluated the stability of our nanoparticles.
Cool. So we have another question from Slido. So the temperature across the nasal cavity varies between 25 to 32 degrees. At what temperature or temperature in your novel polymer gelling after spray in the nasal cavity? And does the nasal cavity torusity affect the gelling process?
Sorry, I cannot really hear you.
Okay, sorry. So the temperature across the nasal cavity varies between 25 degrees to 32.5 degrees, okay? So at what temperature or temperature is your novel polymer gelling after sprayed in the nasal cavity? And does the nasal cavity torusity affect the gelling process?
So in terms of the temperature, at this point, we have looked at 37 degrees, but we studied this previously, and we've showed that I think above 30 degrees, it starts to gellify. Anything below that, it remains as a sort of a liquid formulation. As for the other question in terms of the mucus.
Yeah, the nasal cavity torusity affects the gelling process.
No, it does not affect the gelling process.
I also have a question for you, so I hope you can hear me. So you said that probably your polymer encapsulates the mRNA. Typically, this is based on a very strong electrostatic interaction. Does that work with your polymer, or is it another mechanism?
I think because here there is also a lipofectamine component in it, which is a cationic lipid. So that's so two together combined would generate that nanoparticle system. And because of that functional group, it allows it to interact with the biological component.
Okay. And we have one question over there.
Hi, I just had a question for your biologic nanoparticles. I noticed in the size distribution, there were two different peaks with the measurements from the zeta sizer. Do you have any hypothesis as to what those are? Or is it just a larger PDI?
I mean, I could be, because the nanoparticles are really small, 40 nanometers in size, it could be some aggregation that is happening. Although there are two sort of set of populations, but they are still very small.
And then is that something that, do you feel like those two populations, is that okay for your targets, or is there any concern about that? Or do you think?
No, I don't think there is any concern with that because we also showed that the expression of the EGFP were to a maximal compared to a positive control.
Okay, thank you.
Yeah, so I've got a question myself. Have you ever done stability tests where you took the temperature above the gelling temperature and like raised it above 30 degrees Celsius, or
No,
out of interest?
You mean at 40 degrees?
Oh, 40.
Sorry, did you say that?
So what's the gelling temperature? 30 degrees, right?
Yeah, around
Yeah.
30
Have you done
degrees.
stability tests where you took where you had storage above 30 degrees Celsius and looked at if there's been any impact on your nanoparticles?
The stability?
Yeah.
We are currently doing it now.
Thank you.
So thank you, Wara, again for an
Thank you.
interesting talk.